Okay, so thank you very much for the opportunity to present the work that we're doing at NCI with natural products here. It's a little bit different. You'll see I'll start breaking down complex mixtures into single compounds, but hopefully I'll give plenty of reasoning of why we do that. So, okay, natural products have and continue to be major sources of drugs and drug leads. About 30% of all small molecule drugs are natural product derived. When we look at the when we look at the time period of last 40 years, natural products and their derivatives have represented 50% of all antibacterial drugs, 45% of all antiparasitic drugs, and 40% of all anti-cancer drugs. So they really are important. But when we start having a look at PubMed citations on high throughput screening, we see that it's had an exponential increase till about 2010. Oops. And then, hold on, I want to get my pointer here. Okay, so we're seeing an exponential increase, and then it plateaus to about 2,000 citations a year. But when we want to cross-reference that to natural products, we see that it really stays in the baseline. So we have this treasure trove of chemical diversity and drug potential that's just not getting screened using modern high-throughput screening technologies. So why? Because natural products are complex mixtures. So there's anywhere between 100 to 1,000 small molecules in a natural product extract. And there are some significant challenges associated with screening of natural products. So one is solubility. This is an actual plate that our screening lab calls plate from hell. And you can actually see why. So there's all kinds of solubility issues. There's stuff crashing out of solution, and there's all kinds of color that might interfere with your assay endpoints. Cytotox and off-target issues are another big issue. So here's a kinase kit. So straight out of the kit, if we screen it against a library of natural products, we're going to get a whopping 40% hit rate. This can be improved to give a little bit more responsible hit rate, but it takes a lot of optimization and time to get there. And then finally, natural product isolation is slow. So we follow this lengthy isolation procedure, and we don't know what we have until we're basically done. And so many times, a high-throughput screening assay is over before the structure is determined. And that's a huge problem in screening of natural product libraries, and part of the reason why it stays in the baseline in my high-throughput screening bar graph. So the NCI is home to the Natural Products Repository, and it's one of the largest, most diverse collections of biota used for natural products-based research. Over the last 30 years, the NCI has contracted taxonomists and botanists to go into tropical and subtropical regions of the world where the diversity of the species is highest and collect samples. So our collection is incredibly diverse. We have over 5,500 unique plant genera. Our marine invertebrate collection is represented by over 3,000 unique marine genera, and our fungal collection, which we're now working on, is represented by just under 1,000 unique fungal genera. So, however, there's 200,000 extracts in this library, but the library is that of crude extracts. And so it's associated with all the problems that we talked about in the last few slides, in that nobody really wants to screen it. So to that end, the NCI has started a new initiative called the NCI Program for Natural Product Discovery. It's a joint effort between two different divisions of the NCI, the DCTD and the CCR. There are many aims, and I'm not going to read through them because we're going to go through them in my next couple of slides, but really, sort of the big aim is to reinvigorate natural products-based research and to enable them to be screened against more screening targets than they are now. And so everything is centered on making this NP-MPD prefractionated library. What we wanted to do is we wanted to take those 200,000 extracts from the repository and prefractionate them, go from these complicated mixtures that nobody wants to screen into something that's more screening friendly. And so central to that will be this library of natural product fractions that's amendable to modern high-throughput targeted screening technologies. That library is going to be available in 384 well plates containing a known amount in each well, so 10 micrograms per well of individual samples. Moreover, the library is going to be publicly accessible, free of charge, and open to screening against any disease targets. And so my talk today is going to cover the generation of this library. We're going to start with the methods development and prefractionation. Then we're going to talk about the chemical isolation of compounds from the library. We're going to touch on bioinformatic analysis. And I want to close with an example of an HDS discovery campaign. And so how to start with this 1 million fraction library challenge. We took 16 extracts that represented the repository. So we a couple of different microbes, different marine invertebrates, and then different plants that represented different parts of a plant. And the value proposition was to create methodology where each fraction yields more than 5 milligrams of mass. So we wanted to make enough so that we can support screening for the next 20 years. We had to demonstrate concentration of activity, sort of when crude extract wasn't active, that a fraction was active. And then we also really wanted to demonstrate that we're showing good metabolite separation. If we're going to separate it, we're going to make groupings, polarity groupings of our library. And so we started with these 16 extracts. We looked at 6 different separation media and 2 different solvent systems, one methanol-based, water methanol-based, and the other normal phase based, so more organic solvents. We looked at the mass distribution of all of our fractions. We looked at a 7 and 11 fraction option. And then we screened this entire library in NCI 60. NCI 60, and I will talk about this a little bit further on, is our home assays. There's 60 different cell lines sourced from 9 different panels of human tumors and leukemias. Each bar here you see is individual cell line, and if the bars move from left to right, that means activity. So for example, here we're looking at a crude extract screened at 10 micrograms per mil, which is inactive. And all of a sudden here in fraction 5, we're seeing a single fraction that's active. And that's really one of the key points that we wanted to make. The pre-fractionation brings concentration of activity, where active principles are now a little bit more concentrated in a fraction than they would have been in an extract, and we're picking up activity where we wouldn't have if we were screening crude extracts only. We also looked at metabolic separation. So we ran a lot of LCMS. We did a lot of PCA. We tried to correlate presence, absence of certain ions with activity. We also looked at the concentration of nuisance compounds. Here we're looking at total phenolics in the 7 fractions. And then finally, we wanted to demonstrate that we can go from a mixture to a pure compound in a couple of steps. Something else that was important is to show that we're isolating drug-like molecules. So in the end, we had a look at, there's an online tool called ChemGPSNP that looks at physicochemical properties of natural products, and you can compare them to other groupings of compounds. So here in red, we're looking at compounds that we isolated from our pilot study, and we're comparing them to approved natural product drugs in the last 40 years in gray. And we're really showing that what we are isolating is drug-like and relevant. So in the end, the MP-MPD pre-fractionation methodology takes 250 milligrams of a crude extract and then pre-fractionates it on C8 solid phase media into 7 fractions. The fractions start with 95% water, 5% methanol, and then we're climbing up in methanol, 20, 40, 60, 80, 100% methanol, and a final wash of methanol acetonitrile. Taking 250 milligrams and pre-fractionating on a 2-gram cartridge is a big ask. And so as not to overload, we're actually doing this 2D chromatography where we're actually loading our extract on a pre-cartridge, and then it needs to elute onto the C8 before we start the chromatography. So the pilot study was 16 extracts. We generated a library of about 2,000 fractions, and it took 3 chemists about 6 months to do it by hand. So if we're ever going to solve this 1 million library challenge, we had to automate it. And so our answer to that was a liquid handler. This is a diagram of a typical liquid handler that most labs, including ours, are going to use to transfer liquid from 196 or 300 from one plate onto another. Here, we're going to customize this liquid handler to be able to perform positive pressure chromatography. So it starts with a robot arm and a wash station. We're going to install a light curtain in case anybody's hands go in so that it stops. We're going to be using 7 different solvents. So there's going to be a switching valve that's going to be able to switch between 7 different solvents. There's a central rack that's going to be able to hold our 2D chromatography system. And then the final rack that holds the crude extracts and the C8 SPs. So this is what the design looked like. This is what it looks like in practice. So Here's our two arms, here's our switching valve, here's the rack that holds the extracts and the C8 SPEs, and here's what we're going to start collecting our fractions in. So it starts with equilibrating the column, clipping the crude extracts on, and we're about to start prefractionating. So two arms working in tandem can prefractionate 88 extracts to generate 616 fractions in just under 3 hours. If we do 4 runs a week, we can generate a library of 2,500 fractions a week. And if we operate at 50 weeks a year, we can generate 125,000 fractions a year. So now all of a sudden, this task of generating a library of 1 million fractions doesn't seem so scary anymore. We also made sure that this is reproducible. We took the set of same 88 fractions and then repeated it twice. We had a look at the mass recovery as well as NCI 60 and showed really good correlations with both mass and NCI 60 activity, showing that our prefractionation methodology is reproducible. Things we learned along the way is that nothing was off the shelf. So it ended up being prohibitively long and expensive to actually get a vendor to make equipment for us. So we started making our own equipment. And what really, really helped is getting a 3D printer, because that enabled us to make our own 3D printed labware. So what you're seeing now is a 3D printer making one of these racks that's sort of central to the prefrac that's going to hold the C8 cartridges. It takes about 16 hours and a couple of dollars of plastic to print this. So when we're in full production, we go through about 20 liters of solvent a day that we couldn't just keep under a liquid handler. So we had to install a solvent station in the middle of the lab. It has a drum of methanol, and then water comes in from the building water supply. And it's basically pre-mixing the 7-solvent mixture, and then we had to replumb it to one of our liquid handlers. Sample management, so there's multiple instruments involved here. There's no pretty pictures of sample management, but it's basically endless Excel spreadsheets to make sure that everything's consistent. And then finally, it turned out that 96 well plate looks to a human eye looks the same this way as it does when you turn it 180, but it has disastrous consequences on plate maps. So we started labeling our plates at only a certain corner. We started 3D printing equipment that fits only one way. And we also started running a UV spectra of entire library. For us, this is a quality control measure of plating, but we also found that our screeners found it useful because natural products might absorb UV at wavelengths that can interfere with some assay endpoints. This is a fully automated methodology that involves separation, drying of samples, and then generating plates and then storing fractions in a purpose-built repository. So far, we have made a library of 600,000 fractions. They are plated at 10 micrograms per well in 384 plates, and they're publicly available to screen against any disease targets. We've published methods development and our proof of concept study. And something that was really, really important to us. We found now that we've screened many high throughput discovery campaigns, we find that 70 to 80% of the time, activity was identified in the fraction, but not in the crude extract. And that's really important because it really proves the value of prefractionation. We're seeing where if we were screening crude extracts alone, we would miss on all this activity that we're seeing in the fractions. So now we've moved from this forest full of trees into a single branch. So we've made a simplified natural product sample. And as part of our methods development, we had a look at how many compounds in each one of our fraction. And really the answer is which pair of eyes do you look at it with? So when we look at it with LCMS and ELSD, set the threshold to sort of medium, we can say that each one of our fractions has about 25 to 30 compounds in it. And so this is great because this is now a simplified natural product sample. But really, if we are going to compete with pure compound libraries, we really want to be at a single compound stage. And so the next step had to be methodology that goes from this simplified mixture onto the pure or semi-purified compounds. And just to put things into context, currently there's 500,000 fractions available. If we screen it and get a very responsible hit rate of 0.1%, it's still 500 hits that we need to take to the chemistry lab and go from a mixture to pure compound. So again, everything had to be high capacity and high throughput. So the idea is that the next step Each active fraction is going to be further separated into pure semi-purified compounds. We need to be able to take 500 primary screen hit fractions and process it in 2 weeks' time. So again, throughput. And then we're also going to start getting analytical data so that we can introduce knowledge of structure into this early on. And so our answer to this was to basically take a hit fraction and run HPLC. This is a standardized procedure. So everything's going to be the same. We inject 1 milligram onto a C18 semi-preparative column. So we're using 10 millimeter HPLC columns. We're going to run a standardized gradient that goes from 30% acetonitrile to 100% acetonitrile over 12 minutes. We're going to start fraction collection by time and always by time, and it's always going to be in 30-second increments. So everything's going to be standardized. Quality control. Every 9th injection is going to be this 6-compound standard, and we're going to monitor the retention time of each of these peaks, as well as the difference in the shape of the peak, so that we know exactly how long our column lasts. And finally, we're going to use these high-capacity fraction collectors so that we can introduce throughput into here. And so here's the whole idea of this MP-MPD high-throughput compound ID. We're going to generate these, we call it second stage HPLC plates. We're going to send it back to our screeners in a 2-week period. We're going to get the assay data back. And now we're going to start looking at the chemistry. So dereplication, we're going to run repeat plates. So if something was active in well H4 the first time, we need to make sure that it's active in the same well the second time. And that's why we put so many quality control measures. That's why we run the 6-compound standard every 9th injection. Here, we're going to collect only the active wells, and then we're going to start getting lots and lots of data. So we put value on any kind of data we can get. We routinely run NMR, proton NMR by default, but if signal to noise is good, we're also going to start gathering 2D NMR data, COSY and HSQC in particular. Then we're going to run each one of our active subfractions. We're going to run mass spec on it, high resolution mass spec. And then finally, we're also going to run IR. So get as much data as you want. Following this methodology, we find that in 70% of the time, compound classes can be determined after this single automated subfractionation step. And this significantly reduces chemist time from years for a high throughput screen to 1 to 2 months. And it also reduces the cost of natural product screening and creates timelines that are more amendable to high throughput screening programs. So all of a sudden, chemistry can run with the speed of HTS. Okay. There's something I need to say. So now that we develop methodology for pre-fractionation and second stage HPLC, we start getting a lot of data, and it's both screening data as well as chemistry data. So the next problem in this MP-MPD methods development was, how do we handle all that data? So there's 500,000 fractions available for screening. We run multiple screening campaigns a year. And so there's, as you can imagine, there's a lot of single point data, dose response data, and then analytical chemistry data. And the challenge was, how do you handle that data? And how do you not get lost in it? And so for bioinformatics, I'm going to show you an example of NCI 60. This is our home screening assay. So NCI 60 is 60 cell lines that are representative of 9 different panels of human tumors and leukemia. So each one of these colors is a panel. So red is leukemia, and then each one of these lines is a different cell line. The NCI has been screening natural product extracts for the last 30 years in NCI 60. So there's about 35,000 extracts with 5-dose data, which represents 2 million data points. And so how do you look at it all? How can you look at it all? And there are, okay, so question. And the question is, if I screen something today, how do I know that I have seen this exact dose response profile 5 years ago, 10 years ago, 20 years ago? There are tools that can compare NCI 60 data, such as Compare and CellMiner, but they end up being pairwise comparisons of data, and they're difficult to visualize. Here you're looking at basically 11 by 11. And so again, what I'm aiming to do is look at 35,000 extracts. And so to solve this challenge, we started using self-organizing maps. The value of a self-organizing map is that you can put a lot of data on it, and that it can identify similarity between samples. And so again, this has the potential to solve this question here. Have we seen this dose response profile before? So we've built a neural network or a self-organizing map of NCI60 data. And this is what it looks like. So this neural network is represented by a hexagonal grid of 33 by 19 nodes. The average occupancy is about 10 extracts per node. The smaller the hexagon, the less extracts are in the node. And then similarity, we also wanted to put a dose, we also wanted to assess similarity in this self-organizing map. So the red hexagons are more similar to their neighboring hexagons, the yellow ones are not, and then the blue ones aren't similar to anything else on the map. The way the map is organized, the cytotoxic extracts are generally on the left side of the map, and then the less cytotoxic extracts are on the right side of the map. As we move from right to left, we get a decrease in potency, and as we move from bottom to top, we get an increase in lethal response. The NCI60 SOM is something that is a web-based tool that I can basically parse through from my computer, and you're seeing a small snapshot of what it looks like. And so this enables me to look at 35,000 different extracts all at once. And so this really was, this made a change because it made us believe that we can look at all this data all at once. And so as an example of how we can do this, here I'm mapping all the plant extracts. So every hexagon that you see is actually, there is one plant extract in each one of these nodes. And then I want to map, for example, family Apostinaceae. They're known cardioglycoside producers. And if I want to map all of them onto my map, I can see that there's certain groupings here. Big one here, and big one here. So these are all the extracts of this family on the map. And then, so these Apostinaceae, so these extracts, they have beautiful NCI60 responses. I'm looking at both the potency and then the differential. There's over 2 logs of differential here, and it seems to be very, very potent. This family is well known to make cardioglycosides, and it's the cardioglycosides that are driving the NCI60 response here. And we've proved that by looking at some of the prototypical cardioglycosides and where are they on the map. So when we look at digitoxin, it's right here, and we see that we have a big bunch of extracts in here. When we look at strophanthine, it's here on the map, and again, one of the big lumps of extracts are presented here, as well as nerifolin, which is centered here. I wanted to have a look at, so we heard a lot about different extracts or some botanicals of interest at the meeting today. So I mapped some of them. And most of them, so I have to sort of note that this is a map of cytotoxic extract. And so when I looked at most of them, they're actually not, we may have extracts in the library, but they're not cytotoxic, so they didn't map on the NCI60 SOM. So when I look at the oleander, it shows me 3 small groupings, and oleander is in this family. So it is a cardioglycoside producer, so it fits well to cardioglycosides being what drives the activity here. And then another one that actually shows activity is genus Aristolochia, not as cytotoxic, and so most of it is grouped here. And so this is one way of looking, but this is just looking at biological response. And again, we have a lot more data. So next challenge that we have, and it's not solved, I just want to show you an introduction of what the challenge here is how do you deal with lots and lots of analytical spectra. So we're looking at hundreds of analytical spectra. Here, what I have depicted is just a single 96 well plate of NMR, of proton NMR spectra, of LCMS spectra, and an IR spectra. What we're looking for is to find a method for high throughput data acquisition, processing, and annotation. Each one of these methods has their own advantages and disadvantages. NMR is the most comprehensive. It allows you to see everything with a proton nucleus. However, automated sample preparation is difficult. It's hard to make samples in DMSO that don't get wet using automation. LCMS is very sensitive. We can pick up sort of nanomoles of a compound, but high throughput data acquisition is slow. So even with a UPLC, you need at least about 10 minutes to run a single sample. And then FTIR, which hasn't really been used is recently. FTIR has had a lot of it's gone a long way in the last 10 or 20 years from when I was in grad school. So FTIR became high throughput. And I want to spend a little bit of time sort of plugging FTIR and why it's still useful in the replication of natural products. So FTIR spectroscopy. The value is there's a large spectral range. So whereas in NMR, you're basically looking at 0 to 15. With IR, you're looking at 0 to 4,000. So the spectral range is huge, and that makes it very amenable to library generation and matching. It became high throughput. So when I was an undergrad student, we used to make KBR plates with a big 3-ton press. And now you can run IRs on these silicone plates. So silicone is IR silent. The beam goes right through it. And they make these plate readers. So basically, the plate reader just takes the plate to the IR beam and records it. It takes about 2 minutes to run one IR spectrum. Compare that to about 8 minutes for a proton and 10 minutes for an LCMS, which means that I can record 3 of these plates in 3 IR plates in the same time that I can record 1 NMR and 1 LCMS plate. Short acquisition times and the file sizes are small. So they're really kilobytes. Compare that to an LCMS, high resolution LCMS run that takes a couple of gigabytes. However, there are certain disadvantages in using IR, and really main one is that there's limited natural product spectral libraries available and chemoinformatic tools available. So we're working on that because we're really finding value in working on IR and the dereplication of natural products. So for FTIR libraries, the NCI has worked on natural products for the last 30 years, and we do have pure natural products. So we're making FTIR libraries, and they're all biologically active and isolated from extracts from our repository. We're also, another challenge was nobody really knows how to interpret IR spectra. So this is an IR spectrum with this really simple bromophenol. And other than this stretch here, which I'm going to attribute to this hydroxys here, it's really hard to interpret the rest of the signals. However, we can now use DFT. And so we can compare DFT calculated spectra to the experimental spectra to sort of learn or relearn how to interpret IR spectra. So my last slide on IR is how do we use it? So this is a biochemicals. These are the results of a biochemical screening campaign where we have our fractions on the left. So we're going from 1 to fraction 7, and then we have the subfractions. So the 22 subfractions. If you see the green rectangle, a subfraction was active. And so this was a biochemical screening campaign, and it was all sourced from plants. And we suspected that these early eluting subfractions are tannins. Tannins are a big problem in biochemical screening campaigns. And they're really, the large tannins are actually really hard to see. They're too big to give any meaningful NMR signals, and they're actually too big for the LCMS to pick them up. However, they give beautiful FTIR fingerprints. And when we compare them to our FTIR library, we found that the top library hits were these monomeric units of hydrolyzable tannins. So we find that IR is effective at the identification of tannins, which are common nuisance compounds in biochemical discovery campaigns. It's all done in small scale. So we only used 1 milligram of fraction. We didn't need to scale up. It's done at high throughput and with no extra chromatography steps needed. And for us, it works by giving much better outcomes from plant source projects in biochemical high throughput screening campaigns. And so I want to wrap up on the MP-MPD before we go into the an example of a high throughput screening campaign. So it used to be that we were working on crude natural products, and we went through several iterations of chromatography screening, and natural product drug discovery was really slow. And now we're working on pre-fractionated libraries, and we have this 2-step automated procedure that go from a mixture to a pure compound, thereby allowing natural product science to run at the speed of HTS. So how are we doing so far with MP-MPD? We released, last year, we made our 3rd release, and now there's 500,000 natural product samples available to the public. We've shipped over 8 million wells to screening centers worldwide. On the map here, you're seeing all the screening centers in North America that are screening the library. We've signed 62 MTAs with government, academic, and industrial screening centers, and we've also done technology transfer of MP-MPD methods and automated systems to various groups in the US, as well as South Africa and Sweden. I want to put a disclaimer on the next, for the rest of my talk, I'm going to show an example of high-throughput discovery campaign. However, for most of the discovery campaigns we're actually putting a 3-year embargo on sharing of details or targets, which allows plenty of time to finish the screen, isolate the compounds, and also make sure we can protect these compounds. The one I'm going to present here is collaboration between NCI and NIAID, where we looked at antimicrobial targets. And this is a really, really brief sort of workflow of how the screening went. So we used the first 2 releases of the prefractionated library, which were 326,500 samples sourced from plant, marine, and microbial biota. We did single point screening at 10 micrograms per mil. We took all those hits and did a dose response screen in a 2-fold dilution series. Then we took 75 of the fractions, and we wanted to show that we can go from fraction to pure compound. And in the end, we tested these pure compounds for hit confirmation. So it started with selection of targets. The targets were selected by NIAID, 1 gram positive, 1 gram negative bacterium, as well as a fungus. All 3 of these represent significant threat to human health. And also we found that there's developing resistance to the current drugs used on the market. The NCI provided the screening library, 326,000 fractions. Most of it was sourced from the terrestrial environment, although we had a really good representation of marines. Screening concentration was 10 micrograms per mil in duplicates. The hit rate was a little bit high, but that's because that's because we took a really, really broad definition of what the hit is in one dose. So anything that showed any growth inhibition, we considered a hit here. Then we confirmed these hits in a dose response screen. So this was an 8-dose response screen, and the hit rate was a little bit more responsible, 0.9%, still a little bit high, but I sort of want to say that these are 4 different targets. Most of the hits were antifungal, so against C. albicans, but there was a good representation of E. coli and S. aureus as well. When we have a look at these violin plots, they sort of show the potency. So we were really happy because not everything was here. The violin plots show that we are getting some really, really strong hits at low testing concentrations as well. And so the next step here was to do hit triage and take 75 hits that are representative of diverse activities identified for proof of concept study. Hit selection was based on potency. So the whole idea of this screen is to make it publicly available to encourage further research into antimicrobial natural products. So we took stuff, we didn't take most active hits. We took everything sort of from the top of that violin plot. We looked at all the other screens we did of the MP-MPD, and we selected hits that didn't show any cytotoxicity or activity in other assays. We performed a literature search for taxonomic family and antimicrobial activity. And then we also were cautious of taxonomic redundancy in that we selected only single representative of the genus. And so we ended up with a collection of 75 fractions. Most of them were marines, but also some plants, fungi, and 1 marine alga, red alga. So I'm going to show you an example of a 2nd stage HPLC trace. So here, this was 1 milligram injected onto an HPLC, 22 subfractions collected and tested. The heat map shows activity against the 4 targets, and this was a C. albicans selective hit. We screened both the mother fraction and the 22 subfractions. We're seeing activity in the fraction, as well as these 2 subfractions that are associated with this big HPLC peak. When we started getting analytical data of the peak, this is a proton NMR spectrum done at 128 scans on a 600 megahertz instrument, an IR spectrum, as well as an LCMS. This is plenty of information to replicate this compound. I'm going to remind you that this is done on 1 milligram scale, and we replicated the active principle. Here is this peptide called jaspermide. Using the same methodology, we were able to assign chemotypes to about 70% of our hits. This was done on 1 milligram of fraction material and in about a month of time. So again, faster and effective project selection. From all of that, we took a couple of projects and took it forward, where we isolated the pure compounds and then retested them to confirm activity. One of them was these 2 extracts from Lucetta and Clathrina species sponges. They showed antifungal activity. The active principles were identified to be these 2 aminoimidazoles. I'm showing NMR here because I think it's beautiful. This compound has really low proton to carbon ratio. Proton NMR shows only four resonances, but there's actually a lot more happening than just these 4 proton resonances. There's an internal symmetry in this molecule, so we're seeing even less protons than there is here. When we tested it, the compounds were active and selective against several strains of C. albicans. And what was really interesting is that it was showing activity against azole-resistant C. albicans too. So this 2-aminoimidazole is structurally similar to an azole, and yet it was still showing activity against azole-resistant strains. So this might be a good starting point for looking at antifungal activity of this group of compounds. We also followed some antibacterial leads and isolated these pyridoacridines, so including 2 new deoxyamethidine alkaloids with different bromine substitutions here, and they showed selectivity against S. aureus. In the end, the screen identified around 3,000 fractions with antimicrobial activity. We demonstrated fast and effective active principle isolation protocol, and we also showed the isolation identification of natural products. The results of the high throughput screen are made publicly available for anybody to download, and they're available on our website. Okay, I'm wrapping up here. I think I'm doing good with time. So the MP-MPD has created new opportunities in natural products, new chemical diversity. The prefractionated libraries is publicly available, open to screening against any disease target. We're also working on new collections of cultural organisms. So we're ramping up our fungal collection. New technologies. So we developed technologies for automated prefractionation and rapid compound isolation that can match the speed of HTS. New partnerships. We have collaborations with NCI intramural laboratories, as well as different NIH and various extramural institutes. And also we're isolating new active compounds that are showing cytotoxic, antibacterial, antifungal activity. I'm going to thank everybody I work with. I think our team is amazing because there's a lab full of people that are making robots do chemistry, and that's amazing. And then finally, I'm going to thank all of you for your attention. If anybody's interested in finding out more about MP-MPD, the best person to contact is Natural Products Branch, has an email, and also we have a website that has a lot of useful information of how to contact and work with us. And with that, I'm going to thank you for your attention.
Great. That was really incredible, Tanja. Thank you for walking us through the program, all the analytical challenges, and the great work you've been doing. We have a couple questions already, so I can start with those. But if folks have questions, they can either raise their hand or put it into the chat. Olaf asked, to which extent can the number of fractions be diminished by dereplication? Not sure if I know exactly what that means. Do we need clarification, or do you know what he means?
Yeah, and I think, you know, we don't get any analytical data on the fractions. It's still a little bit too complicated. We have the UV data on the fractions, but you can't really go from UV spectrum of a mixture to a compound. We start getting analytical data when we purify it a little bit more. So dereplicating fractions is hard because there's too many compounds and you don't know what activity is sort of, you know, which compound is responsible for the activity. And that's why we're doing the second stage HPLC, where we go to pure semi-purified compound. And at that point, it's relatively easy. We get as much analytical data as we can. And again in many high throughput screening campaigns, we're successful about 70% of the time of getting to a structure or a chemotype on just 1 milligram. I'm not sure if that answers the question, but I tried. Okay.
Okay. So Olaf gave a thumbs up maybe to his own, so I think so. So Stefan asked maybe, the money question and of how many hits have made it into clinical trials.
Okay, we've been at it for about, I think the first release was about 4 years ago. So we've been at it for only 4 years. It takes a little bit longer than that. Ask me in another couple of years, please.
Stefan, they'd have to do all the safety testing, and that's like 7 or 8 years. No, that's helpful. Could you maybe speak when, you showed the breakdown of the library and the different types like fungal, bacterial, terrestrial? Are you all actively looking for other ones, or how do you maybe add to your library?
We stopped collecting. So we stopped collecting plants and marine invertebrates. What we're really pushing for now is microbes, because they're a little bit more renewable. If we need more, we can just go back to the freezer and get the stock solution. We're working with University of Oklahoma has this citizen science collection program where people collect a little bit of dirt from their backyard. And then the PI there called Professor Chihovich, he actually grows it on Cheerios. Turns out Cheerios are a really good medium to growing fungi because they're full of fat and sugar. And so for the last 5 years, we worked with him, and we got a collection of 24,000 new ITS sequenced fungal samples into the repository. So now we're working on actually growing that up. One part of our lab is this big automation laboratory that does pre-fractionation, and the other part is a fungal lab where we can do 100 1-liter cultures a week. And so we're hoping that in a couple of years, one of the additions to the pre-fractionated library will be this fungal library as well.
Oh, wow. That's incredible. Another thing, when you've worked with people maybe in other countries to obtain these natural products, are they involved after it's been, they get the library and it goes to an external source? Could you speak to that piece?
Yeah, I was thinking of making that slide. So, there's a lot of laws and regulations about going out and collecting. So the NCI operates under the NCI's letter of collection, which basically states that there has to be prior consent from a country before we go collecting. And there has to be, if any of this is ever developed into a drug, there has to be an amicable agreement between whoever develops the drug and the source country. And so before we send our library out, we actually make people sign an MTA that has that. So and there's a specific clause for that, for source country rights. Should you ever develop anything that's commercially, that's a drug, and should you ever start getting any commercial gains from it, you have to go back to the source country and make an agreement about benefit sharing.
That's awesome. Yeah, thanks for speaking to that. Any other questions for Tanja?
I have a message. How often do you combine hits to demonstrate effectiveness versus testing single compounds? Most of the time we test single compounds. The only time I would start combining ingredients is if I lose activity. If at fraction stage I had activity, and then once I separate it into single compounds, I lose activity, or the activity is half, at that point we might be combining, but most of the time we end up testing single compounds.
How often have you seen a loss in effect, or is that pretty rare?
It's rare.
Yeah. Okay. Well, I don't see any more questions or comments. Oh, I'm sorry. Stefan just had one that came through. It seems that plants have been well researched for its chemical constituents. Do you still find novel chemical entities, or is most of it from the marine and fungal sources?
All the time, actually. I come from a marine background, so I like working on marines. I think sometimes they give you a little bit broader chemistry, but I also love working with plants because they tend to be high yielding. We have a lot of plants, so our library is 2/3 plants. And so I know that plants have been thoroughly researched, but we find new chemistry in plants all the time. So keep working on plants. I think that there's big value in working on plants.
Okay. Well, thank you very much, Tanja. Thank you for your elegant presentation and all these great answers to questions.